Okay, so this is a project that I'm just starting. It's my PhD, well, basically my PhD project. I'm in my first year now, and I'm going to look at the population genetics of false codling moth, looking at the gene flow in agricultural environments. Right, so as we all know, the false codling moth is a pest of economic importance, and it affects a wide array of plants. There's been some control measures, which includes insecticides, orchard sanitation, pathogens and biological control agents, as well as mating disruption and the sterile insect technique. The sterile insect technique, or SIT, is a technique of non-lethal sterilization within the pest population, in which sterile individuals are released in the population. All of these practices together do form part of an integrated pest management strategy. Right, so dispersal or movement in the landscape is important, is very important in order to determine the scale of management, as well as to look at the possibility of host races. So knowledge on whether host race is present is important. So host strains are populations that only feed and lay eggs on a particular host or a particular plant species, and thus populations will not persist in other plant species. Pest species specialized on certain hosts are easier to eradicate, as they do not seek refuge in other plants when the host species are out of season. So there is some current knowledge on the dispersal patterns, which is a study that was done by Tim et al. In 2010, and he found that FCM is structured geographically in South Africa. Now, sorry, they only collected fruits of FCM in the Western Cape and the Eastern Cape and Mpumalanga, and they also only sampled a few hosts. And they found that FCM is structured geographically, and that populations within a particular province are more related than those than they are related to different provinces. They also found that FCM is not host specific. However, the study used amplified fragment length polymorphisms, which has less polymorphic information compared to codominant microsatellites. And as I said before, they only sampled from a few provinces and only a few hosts. Right, so the objective of my study is to examine gene flow of FCM in orchard and non-orchards across South Africa, and to examine gene flow within and between major crops or host plants in the Western Cape over 2 to 3 seasons, in order to track individual moths in orchards and home gardens, in order to estimate dispersal and colonization ability between and within populations, and then to determine whether host races exist. Right, so in terms of collection sites for the broad-scale analysis, FCM will be collected from at least 25 georeferenced locations across South Africa in order to investigate movement patterns of FCM. Then in the Western Cape specifically, fruit infested with FCM will be collected from orchards and home gardens, as well as other alternative host plants in a demarcated area in the Western Cape for a period of 2 to 3 seasons. About 10 different host species will be sampled in the area. FCM will be collected from fruits and stored in alcohol for genetic analysis. Microsatellite markers will then be developed. These markers will then be optimized and used for sequencing. Right, so the expected outcomes, the data will be used to analyze population genetic structure in order to understand dispersalability better, and it will also be used to determine whether host races exist. This will aid in control measures, as we will have information on dispersal patterns for the SIT program, and we will know whether host races exist within FCM in order to determine whether populations take refuge in natural and semi-natural vegetation during the off-season. Then I would like to thank the following institutions and persons, and for this project we will need fruit infested with FCM, so if you could possibly help, you can contact me on this email address. Thank you.